Be honest with yourself for a second. How many times have you committed to a serious extended fast, 48 hours, maybe 72, and found yourself somewhere around hour 16, standing in the kitchen, quietly bargaining with your own brain over whether a handful of almonds really counts as breaking it? If that's you, you're in good company. Here's something the fasting world doesn't love to say out loud. Most people simply cannot maintain the kind of prolonged fasting that genuinely drives deep cellular repair. And it isn't a character flaw. It isn't weakness. It's biology doing exactly what biology was built to do: keep you fed. Meanwhile, something interesting has happened. The science underneath all of this has quietly advanced, and a lot of the popular advice hasn't caught up. While the loudest voices online are still romanticizing the suffering of a 3-day water fast, researchers have been mapping out a far more elegant route to the same destination. It turns out there's a specific balance of amino acids, the very building blocks of the protein sitting on your plate tonight, that can throw the same internal switch along fast throws. Same biological result, a completely different road to get there. No starvation, no crashing energy, no mental fog so heavy you forget what you walked into the room to do. Just your body quietly cleaning house from the inside, the way it was always designed to. Stick with me, because by the end of this, your dinner is going to look like a control panel. Start with a simple question. When you fast, what do you actually think is doing the work? Most people would answer, not eating, resting the gut, burning fat instead of sugar. All real, all true, and all surface level. The mechanism that has longevity scientists genuinely excited has very little to do with calories. It has everything to do with signals, tiny chemical messages that your cells are constantly receiving and reacting to. And one of the most powerful reactions those messages can trigger has a name: autophagy. The word sounds intimidating, but the idea is almost beautiful once it clicks. Auto means self, phagy means eating. Your cells eat themselves, but not in any frightening sense. Picture each cell as a working garage. Over time, that garage fills with clutter, broken tools, dead equipment, parts that no longer function and just take up room. Autophagy is the maintenance crew. It walks in, identifies everything worn out or defective, and recycles it, breaking it down, reusing what's useful, hauling away the rest. When that crew is allowed to do its job, you think sharper, you age slower, your immune defenses run better, and your mitochondria, the tiny power plants inside every cell, run cleaner and more efficiently. This isn't fringe territory. A Japanese cell biologist named Yoshinori Osumi spent decades decoding precisely how this process operates at the molecular level, and in 2016 that work earned him the Nobel Prize in Medicine. So when we talk about autophagy, we're not talking about a wellness trend. We're talking about decades of peer-reviewed, prize-winning science. Now here's the part that rewrites the old story. For a long stretch, researchers assumed autophagy had exactly one trigger: total caloric deprivation. Stop eating, full stop. And that belief looked airtight. Fasted animals showed elevated autophagy, fed animals showed it drop. Clean and logical. But as the tools got sharper, scientists stopped asking only whether an animal was eating and started asking what it was eating. And the picture changed. It wasn't the absence of calories switching autophagy on, it was the absence of certain molecular signals, and those signals are carried by specific amino acids. That single distinction is the crack in the wall that lets the light through, because if you can control the signal, you no longer have to control the calories. So let's meet the molecule that may, right now, be standing between you and your body's repair system. Even if you eat clean, train hard, sleep well, and do everything else right. It's called mTOR, mechanistic target of rapamycin. Forget the name, focus on the job. mTOR is your cellular site manager. When it's switched on, it tells the body, we're building, synthesize, accumulate, grow. Muscle gets assembled, proteins get manufactured, everything points toward expansion. When mTOR switches off, the manager clocks out. And only then does the cleanup crew, autophagy, finally get to work. This is the crucial rule. Growth mode and repair mode cannot run at the same time. It's always one or the other. And your body makes that choice constantly, based on the signals it reads. One amino acid, more than any other, is the master switch telling mTOR to turn on: leucine. Leucine is a branched-chain amino acid, and it saturates the standard Western diet: chicken breast, beef, eggs, dairy, whey protein. Your body is extraordinarily sensitive to it. Research published in Nature Metabolism found that even modest amounts of leucine, well under what a normal high-protein meal delivers, are enough to fully activate mTOR signaling in liver and muscle. Sit with that. The protein shake you drank this morning for recovery didn't only feed your muscles, it also slammed the door on the cleanup crew that had been repairing damage all night. Leucine doesn't act alone. Its branched-chain relatives, isoleucine and valine, push mTOR the same direction with less force. And there's another character worth knowing: methionine, a sulfur-containing amino acid concentrated in animal muscle, beef especially, but also poultry and eggs. Methionine fires up a separate but equally effective autophagy blocker called IGF-1, insulin-like growth factor 1. Think of it as a second deadbolt on the same door. mTOR is lock 1. IGF-1 is lock 2. A diet heavy in leucine and methionine keeps both engaged. And here's what even careful, health-conscious eaters miss: you don't have to abandon protein to release those locks. You just have to change which proteins and in what proportions. That's the loophole nobody talks about. So here's the actual formula. Your cells run internal sensors, systems with names like AMPK and GCN2, that continuously sample the amino acids circulating in your blood. They aren't just measuring how much protein you ate, they're reading what kind and in what ratio. Specifically, they track the balance between essential amino acids, the ones you must get from food, and non-essential ones, which your body can produce itself. When that balance tips hard toward essential amino acids, the way it does after a steak, the sensors read abundance. mTOR fires, autophagy stops. When non-essential amino acids make up the larger share, the sensors read something closer to scarcity. And scarcity is the language autophagy understands. Work from the Salk Institute, supported by longevity research out of USC, has helped sharpen where the threshold sits. When non-essential amino acids make up roughly 60 to 70% of your total intake, with leucine held under about 8% and methionine under about 5%, your body can enter and hold an autophagy-favorable state with no caloric restriction at all. You're eating. You're satisfied. And your cells behave as though you're fasting. Now, the methionine piece answers the fear everyone raises the moment they hear restrict an amino acid. Won't I lose muscle? Reasonable worry, but here's what the research shows. Methionine and cysteine, another sulfur-based amino acid, share a metabolic pathway. The body can convert between them. When you lower methionine while keeping cysteine adequate, you still get the drop in IGF-1 and the quieting of mTOR, but you avoid the muscle loss that blunt methionine restriction causes. It's like finding the one loose thread that unravels only the part of the sweater you wanted gone. A study in Cell Metabolism showed mice on a low methionine, adequate cysteine diet displayed autophagy markers on par with animals fasted for 36 hours. 36 hours of fasting without skipping a single meal. There's one more elegant idea here, sometimes called the dilution effect. Certain plant proteins, hemp seeds, spirulina, particular legume pairings, carry naturally low leucine and high proportions of non-essential amino acids. When those proteins dominate your plate, a sensing pathway called SESN2, which reports straight to mTOR, fails to register abundance. It registers scarcity instead. The gate opens, the cleanup begins. All while you're sitting down to a genuine meal. So what does this look like on an ordinary Tuesday? Here's a flexible 24-hour structure. Adapt it to your life, not the reverse. In the morning, you simply extend what's already happening. After 7 to 9 hours of overnight sleep, you wake up in a mild autophagy state already. The crew has been working. Your only job is not to shut the door before they finish. That means holding off on leucine-dense protein first thing. No eggs, no whey, no Greek yogurt yet. What you can have: black coffee, which independently activates AMPK and is genuinely useful here, not just for the caffeine. Green tea, whose catechin EGCG acts as a mild mTOR inhibitor. Fruit, non-starchy vegetables, and a small amount of hemp seeds, or spirulina if you want protein without spoiling the ratio. You're not white-knuckling a fast. You're eating strategically. Your midday meal is the bridge, and it's where the amino acid ratio does the heavy lifting. Build it around plant proteins with favorable profiles, lentils and rice, which alone are incomplete, but together complement each other while keeping leucine well below an equivalent serving of chicken. Hemp seeds, sitting around 6% leucine, right in the target zone. Pumpkin seeds, rich in non-essential amino acids like glycine, which may actually support autophagy signaling. Chickpeas with tahini, favorable. And it genuinely tastes good, which matters more than the health world likes to admit. Pour on olive oil and pile on non-starchy vegetables. Fat doesn't activate mTOR, and vegetables don't trip the sensors. You'll be full while your cells keep cleaning. The evening meal is the strategic reload, and here it gets counterintuitive. This is where you deliberately bring back higher quality, higher leucine protein. The logic is simple sequencing. You spent the day protecting the cleanup state, so now you hand the construction crew its shift. Muscle protein synthesis runs mostly overnight during sleep, so loading quality protein in the evening lines your biology up cleanly. Repair by day, rebuild by night. The ideal choice is fish, salmon, mackerel, sardines. Because the amino acid profile is excellent, the methionine is meaningfully lower than beef, and the omega-3s fight inflammation on their own. Pair it with complex carbohydrates like sweet potato or quinoa to soften the insulin response and lower overnight cortisol. The food protocol alone is enough, but if you want to deepen the signal, 3 compounds have real mechanistic backing. Spermidine, found in wheat germ and aged cheese, induces autophagy through a pathway independent of mTOR. It works even when mTOR isn't fully off. Urolithin A, produced when gut bacteria process compounds from pomegranate and certain berries, specifically triggers mitophagy, the recycling of damaged mitochondria. Berberine, a well-studied AMPK activator, mimics several of the signaling effects of caloric restriction. None of these are magic, and none replace the diet. But they compound the signal in the right direction. As for timing, here's what the research honestly shows. Through the first week, the shift is quiet and mostly under the surface. Your nutrient-sensing pathways recalibrate, autophagy flux rises, and mitochondrial quality control begins. From roughly day 8 to 21, you start to feel it. The afternoon energy crash eases as mitochondria run cleaner. Skin texture often improves since skin cells are highly autophagy active, and inflammation markers like IL-6 and CRP tend to drift downward. By day 22 to 30, studies on autophagy-supportive eating consistently show broader gains. Improved insulin sensitivity, sharper cognition, as autophagy-dependent neurons run better, and often deeper sleep. And you don't have to take any of it on faith. You can measure it. The glucose ketone index, or GKI, remains a reliable indirect marker. A reading under 3.0 correlates strongly with active autophagy, and a continuous glucose monitor paired with a ketone meter gives you that in real time. An IGF-1 blood panel, widely available through direct-to-consumer labs, directly tracks one of the 2 main autophagy-blocking hormones. You want it sitting in the lower normal range. And specialized functional medicine labs now measure autophagy proteins like bclin-1 and LC3B directly. Track these over 30 days, and you're working from data, not a guess. So let's bring it home. Your body carries a sophisticated, Nobel-validated repair system in every cell you own. For decades, we assumed the only key was to stop eating entirely. But the real switch isn't calories. It's signals. And the strongest signals come from the ratio and composition of the amino acids your body detects. High leucine, high methionine, growth mode on, autophagy door shut. Low leucine, restricted methionine, non-essential amino acids dominant, mTOR quiets, AMPK takes over, and the door swings open. In practice, extend your morning window with coffee, green tea, and low-leucine foods. Build midday around plant proteins that hit the favorable ratio. Reload in the evening with fish to drive overnight repair. Layer in spermidine, berberine, or urolithin A if you want to push further. And track your GKI and IGF-1 so you're guided by data, not a feeling. That's the longevity payoff of chronic fasting. Without the cortisol, without the muscle loss, without canceling dinner because you're on day 2 of a water fast. There's one more layer most people completely overlook. Your circadian rhythm directly shapes how efficient your autophagy actually is. The exact same meal eaten at different hours of the day produces strikingly different cellular outcomes. Same food, different clock, different biology. That's exactly where we're going next time, so make sure you don't miss it.